Okay, so we did carbohydrate digestion, and now we want to move on and look at the other components that you bring into your digestive system and see how they digest as well. So we are going to start with protein digestion, which would require that you would digest a protein, actually ingest a protein into your mouth. A protein is basically, if you remember the pop beads that you had when you were a kid, a protein is made up of a string of these pop beads. Each little pop bead is an amino acid. Okay, and they're made up of thousands of these different amino acids, and we get different proteins by arranging different amino acids in different orders and so forth and so on. So it comes into the mouth, and protein digestion doesn't begin until we get down into the stomach. Once we're in the stomach, there are two cells in the lining of the stomach that produce things that are going to help. The one cell is going to produce hydrochloric acid, okay, and the other one is going to produce pepsinogen. Pepsinogen is a zymogen. Let's respell that one. Is a zymogen. And a zymogen is a precursor to an enzyme. So it's sometimes it's referred to as proenzymes as well. So these are enzymes that are going to they're formed, but they are not active. So it's an inactive enzyme. When pepsinogen comes in the presence of hydrochloric acid, that hydrochloric acid is going to convert it into pepsin, and this is now the active form of that enzyme, and it's going to work on the proteins that were coming in here, and some of these proteins will begin to be broken down into polypeptides in the stomach, but not all of them. So leaving the stomach, we'll have some polypeptides, we'll have some proteins, and they're going to enter the duodenum. In the pancreas, there are two enzymes that are going to act on the polypeptides and proteins that are coming from the stomach. Trypsin is going to act on the proteins and break those down. And then we'll have a bunch of different peptidases, and I'm not going to ask you to name specific ones. I just simply want you to know that there are peptidases that'll break down those polypeptides. Also, here in the small intestine, on that brush border that we talked about in the carbohydrate digestion, are other peptidases. And again, I could go through, we can name these specifically, but I'm going to choose not to. The peptidases are going to work on these polypeptides, break them down into smaller and smaller pieces, until eventually it's broken down into its monomer, which are these amino acids. Okay, we talked about monomers in the carbohydrate digestion. This is the smallest unit that I can break a substance down into, and this is a unit that it needs to be broken down into in order for this to be absorbed. So we need this monomer to actually absorb the pieces of protein into our system. And so the amino acids are going to go into the digestive capillary beds, and then from there, from these digestive, it'll go into the digestive capillary beds, and from the digestive capillary beds, they'll go to the liver, and from the liver, they will enter the main bloodstream. So protein digestion is not nearly as complicated as what the carbohydrate digestion was. Relatively straightforward. Start with proteins, use either pepsin or trypsin to break the proteins down into polypeptides, and then the peptidases are going to break those polypeptides down into amino acids, and then the amino acids are going to be absorbed into the capillary beds, which will take it into the bloodstream. So now let's look at fats. And fats have a little bit different, lipids have a little bit different process. So let's take this one away. And now we're going to start over again, and we're going to do lipid digestion. When we look at a lipid is made up of a single, let's see where you can see, a single monoglyceride and attached to the monoglyceride are 3 fatty acids. Okay, so it's kind of looks like a key or something along that line. So it's one big monoglyceride, and then it has 3 fatty acid tails. And if you look on the back of a food label and you see saturated and unsaturated fats, it has to do with the structure of these fatty acids. We're not going to get into the details of that today, but just wanted you to be aware of that. So the lipid enters the mouth, and there may be some of us produce a little bit of something called lingual lipase, which may begin to break down the lipids. It's not overly effective, and not everybody makes that particular enzyme, but it is there. So it's produced in the oral cavity. It's going to start the breakdown of lipids in the oral cavity, but pretty much lipid digestion really begins after it passes through the stomach and once it gets into the small intestine. And then once it gets into this small intestine, it's a 2-part process. So this lipid, the first thing that has to happen, and let me give you an idea. So if you look at a lipid and we think about it as being this big drop, so this is a big drop of fat, okay? So it's these monoglycerides and fatty acids that we talked about up here. The first thing we need to do, because you all know fats, waters, they don't mix, I need to be able to get more surface area here for my enzymes to be able to work on. And the easiest way to do that is to break this fat droplet down into smaller pieces. And so when bile is present, okay, we're going to put bile here, and when bile is present, it doesn't chemically digest the fat, but it causes this fat droplet to break down into a whole bunch of smaller drops of fat, okay? And so now, instead of having a very small surface area, I have increased my surface area significantly. This is called emulsified fat, okay? It is not a chemical change. And that's what an enzyme would do. An enzyme would make a chemical change. It is a physical change only, because we are going from a big drop of fat to small drops of fat. Okay, it's still fat, it's just a different size. So bile is not an enzyme. You can be pretty sure that I'm going to ask a question similar to that on an exam. Can you explain to me why bile is not an enzyme? It's important to the digestive process, because if we don't have bile, it's very difficult to get enough surface area to properly digest those fats. But it doesn't do a chemical change. It only does a physical change. So bile is stored here in the gallbladder. All right, there's where you find bile, and it comes out this little duct and into the duodenum. And then here in the pancreas, we have an enzyme called lipase, and it comes out as well. And so the bile is going to turn this lipid into an emulsified fat. Okay, so it's going to turn it into an emulsified fat. And the lipase is going to break this emulsified fat down into its monomers, which are monoglycerides and fatty acids. Okay, and so here are the monomers for a lipid. Okay, monoglycerides and fatty acids. Good test question as well. Tell me what the monomers are for each of the nutrients that we're breaking down. So carbohydrate, the monomer is a monosaccharide like glucose. In proteins, the monomer is amino acids. In lipids, the monomers are monoglycerides and fatty acids. These monoglycerides and fatty acids are going to move out of the capillary beds, and they're going to move into a structure called a lacteal. Okay, and this is the lymphatic, it's a lymphatic structure that you're going to find in a microvilli. So if we look at a microvilli, remember it's on the surface of those columnar cells. We're going to enlarge that microvilli, and in the center of that microvilli we have a lacteal, and it's basically a tube that comes in there. And then surrounding that lacteal, let's label that. So this is a lacteal. So now you can see the lacteal and the microvilli are labeled, and the monoglyceride and fatty acids will enter that lacteal. And if you remember, the lymphatic system eventually does end up depositing that material into the circulatory system up in the subclavian veins, and that's how these get into the circulatory system. All right, that's the end of both protein and lipid digestion. The last video will go over nucleic acid and the assignment that goes along with this. Thanks.